Right. Now we can start. Yeah, so today we want to talk about the effect of pesticides when they enter the plant system or insect pest system, or they could interact with the pathogens. So this is what we want to talk about. We want to find out what will happen to these pesticides. It may also help us to understand about pesticide resistance. So let me start sharing. So our topic is pesticide metabolism. Now we know the pesticide can enter the animal system. Later on, there will be enzymes in the animal system which will cause biotransformations, metabolism or co-metabolism, and finally there's excretion of the products. In the plant, the same processes could also take place, but they could be in cooperation in the plants. Then there's also microbial activity. We can divide the pesticide metabolism in 3 phases. Phase 1, phase 2, and phase 3. Now, in phase 1, normally we have the formation of these functional groups. Just give you a few examples. The hydroxyl OH, the SH, the carboxyl COOH, the cyano group, CN, then the amine group. So we can have our pesticides forming these radicals. Now in phase 2, we may have glutathione, glucose, but in animals we may also have a glucuronic acid instead of glucose. We have these conjugation reactions taking place, and also we may have amino acid conjugations. Now in phase 3, we could have further conjugation in in plants. So we want to look at the fate of pesticide or pesticides inside a living system. In other words, what is going to happen to the pesticide when it enters the living organism? The pesticide could participate in metabolism or co-metabolism. By co-metabolism, we mean that transformation makes pesticides less toxic to target or non-target organisms. So when the pesticide has entered the living system, there are changes which are going to take place to make it less toxic. And these are some of the reactions which are likely to take place. First, we've got oxidation. Second, we've got reduction. Third, we've got hydrolysis. And the fourth process is conjugation. We could also have the biotransformation of an organic compound that is not used as an energy source or a constructive element of the organism. Nevertheless, changes will take place in that organic compound. Now, in order to facilitate the changes or chemical breakdown of pesticides, living systems are endowed with the enzymes. We have got different classes of these enzymes. Number one, we have got what are known as hydrolytic enzymes. There are three of them: esterases, amidases, and nitrases. Second, we have got the oxidases, where we have cytochrome P450s and the peroxidases. And we also have reductases. Reductases, we have got nitro reductases, and we have got reductive nitro reductases. Then we've got another group, transferases, glutathione S transferases, and the glycosylation. We talk about these processes. We are going to mention some of these enzymes so that you know where exactly they become useful. Now, if we look at the cells of organisms, they contain enzymes such as the ones which we have mentioned. These enzymes are capable of oxidizing pesticides. For example, we have got microsomal enzymes that are responsible for pesticide oxidation, microsomal oxidases, or mixed function oxidases. I think here we are mainly talking about the cytochrome P450. We'll talk about it more. Enzymes in the cytosol are able to oxidize certain alcohols and the aldehydes. Reduction processes are also done by some microsomal enzymes. Nitro groups could be reduced to amino groups. Now, conjugation is facilitated by a membrane-bound enzyme, glucuronic acid conjugates are common in higher animals. So in higher animals, we have glucuronic acid conjugations, but in plants, we mainly conjugations. Conjugating substances are water soluble. Now, let us focus on phase one processes. Here we have got hydrolysis. These are the enzymes which can break down pesticides that contain ester, amide, or phosphate linkages. So you need ester, amide, or phosphate to have hydrolysis to facilitate the breakdown of the pesticides. Now, the examples of insecticides which are prone to hydrolysis, we have got organophosphorus, pyrethroids, carbamates. These can also be broken by hydrolysis. The fungicides, we have got dithiocarbamates, dinitrophenol. These can also succumb to hydrolysis. Then we have got the herbicides, urea and carbamates. They can also be broken down by hydrolysis. So hydrolysis are present in all living organisms. Now, organophosphorus contain the ROP linkages. And the ROP linkages are attacked by hydrolysis. And probably we could have special names for these hydrolyses. For those ones which specialize on ROP linkages, we may call them phosphatases. Another group of hydrolyses which operate on the amide. In amide is where you have a CO attached to N. The enzymes or the hydrolyses which affect this group are known as carboxylamidases. Carboxylamidases. You could be aware of malathion dust. This is an insecticide. This malathion is actually an ester. It's formulated as an ester. And it can be changed to an acid and an alcohol through the carboxylase enzyme. So malathion is efficiently converted to acid and alcohol components in higher animals. Probably this is the reason why it is not very toxic to higher animals. But of course, it's quite toxic to insects. But the difference is that in higher animals, malathion can be broken down very fast by carboxylases or hydrolysis. So we have these hydrolysis. So this ester is attacked by the carboxylase. We also have the hydrolysis of esters of 2,4-D in plants. We know 2,4-D is a herbicide. Normally, we apply it to plants in the form of an ester to facilitate its entry into the plant. But once it is inside the plant, we want it to release the acid part, which is active dichlorophenoxyacetic acid 2,4-D, which kills the weeds. So hydrolysis can actually liberate the acid part of the herbicide. And many pesticides may contain the OCH3 or the OC2H5 alkoxy groups, which can be broken down by hydrolysis. Amides. Amides are attacked by special hydrolysis known as amidases. And for our example, we are going to use dimethoate. This is the structure of dimethoate. It's an organophosphate which has got a direct S sulfur here, which is attached to this amide group. So we have amidases, which are going to break this amide to form an acid and an amine. So when we spray dimethoate, probably what is going to happen in plants or aphids is that it is going to be changed to an acid part and an amine part through hydrolysis or through this hydrolysis. For example, amidase. The same thing can happen also to the herbicide propanil in rice, which is attacked by carboxyl esterases. It's carboxyl, I think there's L here, yeah. Carboxyl esterases, which can attack amides. So this is our propanil which is split into this part here. Then it forms this acid plus this bit of the pesticide. Then we have got Cabril. Now, Cabril may form what is known as an epoxide. And we have got the enzyme epoxide hydrolase. Or the enzyme may be called hydrotase. Or hydrase. But probably let us just stick with epoxide hydrolase. So this is the structure of Cabril. Now, somewhere here it is going to open up and form an epoxide. Now, after forming that epoxide, a further reaction will convert the epoxide to dihydrodiol. It's a dihydrodiol where we have the two hydroxyl groups now being attached to this structure. So epoxide hydrolase can actually change the structure of Cabril. It becomes less toxic it could be in the insect or plant. Still on the phase 1 processes, probably we can put on a summary about the active enzymes here, where we have hydrolases. If we have such a structure, then we have got carboxyl esterase, which can act on this structure. It is still a hydrolase. Then if we have got an amide, we have got an amidase. Again, here it's an amide, which is an amidase. And these could be the products. Now, we have talked about the role of hydrolases in facilitating the chemical reactions which can take place on the pesticides. We also have Cytochrome P450 enzyme, which can facilitate mainly oxidation reactions. But in phase 1, we will have oxidation reactions, hydrolysis, reduction, and actually these three, oxidation, hydrolysis, and reduction, are occurring in phase 1. And the conjugation is occurring in phase 2 and 3. So the breakdown of pesticides is mostly facilitated via three phases. We have already talked about the activity of hydrolysis in phase 1. There are also other reactions in phase 1, which are oxidation, reduction, and hydrolysis. Then in phase 2, we have conjugation. Phase 3, we can still have further conjugation, and the conjugates are now moved to the vacuoles if it is in a plant, or to the cell walls. But if it is in the insects, after phase 2 conjugation, then there is excretion of the metabolites. Now let us now focus on cytochrome P450 monoxinase, which is the enzyme which facilitates breakdown of chemicals or pesticides in living systems. Now, cytochrome P450 resides in the endoplasmic reticulum in the cells. It can form what is known as the monooxygenase system because it adds oxygen to the pesticides. Sometimes the same enzyme is known as mixed function oxidase or microsomal oxidase. Basically, the enzyme catalyzes oxidation reactions. In this case, oxygen is incorporated into the pesticide molecule to cause some changes. The oxygen atom is converted into water by the electron donor NADPH. This then is the basic reaction of cytochrome P450, where you have NADPH plus H plus and oxygen plus RH equal to NAD plus plus H2O plus ROH. So your RH represents the pesticide here, and ROH represents the altered pesticide metabolite. So this is the pesticide metabolite, the product of oxidation or a changed pesticide. Please note that it's not only pesticides which undergo these changes. There could be other foreign bodies apart from pesticides, but all substances that enter the living system, we refer to them as xenobiotics or just simply foreign substances. Now, when the pesticide enters the living system, what is happening is that the pesticide is altered by the addition of functional groups, where you have the OH, NH2, SH, it could be COOH, CN, OCH3, CH3, and C and this SCH3. These functional groups could be added to either herbicide molecule, insecticide, or a fungicide, which will reduce the toxicity. Maybe let us change here to pesticide. pesticide molecule. Now, when the pesticide has entered the living system, under the influence of cytochrome P450, there's a possibility of 7 possible reactions. This is in addition to the activity of hydrolysis. I mean, with hydrolysis, we have seen their activity on amides and esters. And we gave the hydrolysis special names like amidases or carboxyl esterases. But this now is the activity of cytochrome P450, another enzyme, facilitating at least 7 reactions. The first one is hydroxylation. Second is N-dealkylation. Third, O-dealkylation. 4, oxidative deamination. 5, sulfoxidation or thioether oxidation. And 6, epoxidation. Number 7, we have got the replacement of sulfur atom by oxygen. So let us examine hydroxylation, what is actually happening. You could have a pesticide carrying a chlorine atom. This is substituted by an OH group or hydroxyl group under the influence of cytochrome P450. And this whole process becomes hydroxylation. This could be from the aromatic group because of the benzene ring. If it is a straight chain carbon atom group or aliphatic group, we also have the replacement of Cl plus OH. Again, hydroxylation. Here we have got an insecticide known as pyrethrin, which is going to undergo hydroxylation. It can happen here. So you can have hydroxylation taking place where you have the carbon atom. So that is an insecticide, pyrethrin. Hydroxylation can also take place on a herbicide, Basagran. So on the benzene ring, we have got an insertion of the OH group during hydroxylation under the influence of cytochrome P450. Now, if we spray Basagran to soybean, soybean can change Basagran by adding an OH to the molecule of Basagran. And this is how Basagran becomes detoxified via the process of hydroxylation. Carbaryl is an insecticide. It can also be hydroxylated. You can see this is the molecule of carbaryl. After hydroxylation, through the influence of cytochrome P450, we can add OH either here or there. There are 2 possible positions where we can insert the hydroxyl group. Once we have done that, then carbaryl becomes less toxic. So that is the example. Those are the examples of hydroxylation. Addition of the hydroxyl group to the either insecticide or herbicide to deactivate the molecule. Then there's what we call N-dealkylation. This is the removal of this group. It could be the methyl group attached to nitrogen and to the rest of the molecule. Then we end up having an H2. This can happen to atrazine. Here you can have, this is the structure of atrazine where we are having this N, and we can convert this to an amine through probably cytochrome P450, although another enzyme is now known to facilitate this. We can simplify this bit, the N-dealkylation, so that we create an amine. Instead of having cytochrome P450, it's now being thought that an enzyme known as DIMBOA is now responsible for these changes, or dealkylation process in atrazine. But in other compounds, cytochrome P450 is responsible. Now we also have diuron, which is it's a herbicide where we can actually remove CH3 here. That is the process of dealkylation, and add an H there. It's known as N-dealkylation. There's also what we call O-dealkylation. If we have this structure here where we have got R attached to OCH3, we can remove this methyl group and remain with an OH there. So this is the basic reaction, very simplified, so that you know where the changes are taking place. Instead of focusing on the complex molecule, we are just focusing on something which is simple. This is 2,4-D acid. Breakdown or dealkylation can take place here, where we can remove this whole bit and replace it with an OH. This is actually making this 2,4-D less toxic. Methochlor, I think this is an insecticide. Now, what can happen is that we can do O-dealkylation here at this position here, CH3, remove the methyl group so that we are remaining with an OH, and we are making this pesticide less toxic. This then is what is known as sulfoxidation or thioether oxidation. Now, when you have a sulfur within the molecule of a pesticide, there's a possibility of oxygen combining with sulfur through a process known as sulfoxidation or thioether oxidation. And this is under the influence of cytochrome P450. So if we just take this simple structure, we just add oxygen here, and the process becomes sulfoxidation or thioether oxidation. Here we have got the prometrin, which is the herbicide with this structure. Where we are worried about is where we get S. If we add oxygen to the S, then we have deactivated prometrin. It's no longer an active herbicide through that process of sulfoxidation. And by the way, such a process can be catalyzed by cytochrome P450. Here, I also want to illustrate something to do with sulfoxidation or thioether oxidation on the herbicide known as EPTC, which is used to kill cyperus, either in maize. Now, EPTC happens to have this S. So we'll add oxygen where there's S, and the process is known as sulfoxidation or thioether oxidation, and this is under the influence of cytochrome P450. We can have a simplified version of sulfoxidation here, where we have put a circle around where we find S, then we put oxygen where we have this S, and we would have deactivated that herbicide. The other process is known as oxidative deamination. Now, if we are going to have this process, we should have two nitrogen atoms which are joined together, and that should occur somewhere in a molecule which is likely to undergo oxidative deamination. What it simply means is that this other nitrogen is going to be removed. Then we are going to remain with one nitrogen attached to hydrogen. This then is oxidative deamination using metribuzin as an example. We have got this double where we have got nitrogen here attached to nitrogen there. So this can be removed. Then we are left with one nitrogen there as a result of oxidative deamination. This can occur in metribuzin or sengo, which is the herbicide used to control weeds in soybean. Nevertheless, if we have any pesticide for that matter, having such a structure, that pesticide is likely to undergo a similar reaction, oxidative deamination. Remember, there are several of these pesticides. We only need to identify whether we have nitrogen attached to another nitrogen. Probably by now you should know that if you have got S here representing sulfur, probably apart from oxidative deamination, this can also undergo sulfoxidation or thioether oxidation. Here we are simply adding oxygen to the sulfur. It's another way of deactivating metribuzin. Then we've got this process think hydrolyses are involved here. With the reduction, we need the nitro group, which is converted to an amine. For example, like on this herbicide, trifluorine with these nitro groups, two nitro groups, which can actually be converted to an amine. This is a nitro reduction. We can simplify it like this, where we have the nitro group being converted to an amine. The other reaction is known as epoxidation. Now, epoxidation, we have such a structure. Now, what is going to happen is that we simply add oxygen there. Then that becomes an epoxide. For example, here, where we have this is aldrin. It's an insecticide, and we can affect this portion here through oxidation and add oxygen. Once we have done this, this becomes an epoxide. The epoxide is now dieldrin. But after epoxidation of aldrin, it doesn't mean dieldrin becomes less toxic. Actually, it becomes very toxic. So sometimes the change can lead to a very toxic substance. We also have a replacement of sulfur atom by oxygen, where you have the organophosphate. This is not sulfoxidation, but simply a replacement of sulfur by oxygen. For example, we have got the insecticide known as parathion. This is parathion. I think we can make it even more toxic by removing S and replace S with the oxygen. So parathion becomes paraxone. And it's quite toxic. Now we move into hydrolysis, which can also cause some changes to the pesticides. Now there are three things which can happen when we add water to the pesticide, there's going to be a reaction with water. But water can only react with certain types of pesticides. These pesticides should possess, number one, they should have an ester. They should have a nitrile group, or they should have an amide group. So they should be ester, nitrile, or an amide. So if we add water to such pesticides, they will degrade or they will undergo a reaction. This is what we are going to examine. First, we are going to look at the ester hydrolysis. So the key issue is that there should be an ester structure on the pesticide for it to react with water. After reacting with water, it forms an acid plus an alcohol. So let us check some of the pesticides we have. We can have 2,4-D-methyl ester having this structure here. If we add water to it, it will break down into an acid, 2,4 acid plus alcohol. And the whole process, we call it de-esterification. Remember, sometimes we deliberately form a methyl ester of 2,4-D to facilitate its entry into a plant. But once in the plant, we want it to break down via de-esterification so that the acid is released so that it can start to kill the weed. The other one is we should have an amide, then we have amide hydrolysis, or if we add water to an amide, we get hydrolysis, which liberates an amine. So this is the structure of the amide. After the addition of water, we get the amine. We can use the example of propanil, which is the herbicide having this amide. Then the addition of water will cause it to form an amine here plus an acid. So please note that we could have other pesticides having such a structure, and these pesticides are likely to undergo the same fate. Here we've got the cyano group, which is carbon attached to nitrogen via a triple bond. So what happens is that the cyano group, sorry. Hello? Tiripo. Yeah, right now I've got a lecture. Okay. Okay, sorry about the disturbance. So here I was going to talk about the nitrile hydrolysis. where we change it first to an amide, then from an amide, it goes to an acid plus an amine. We have an example of cyanazine or bladex, where we have this triple bond here. It's converted to an amide. Then again, the amide is converted to an acid. Again, this can occur on the bromoxynil bacterial wheat herbicide, which is also converted to an acid plus ammonia. By the way, we could have fungicides and other pesticides, as long as they possess this cyano group, they are likely to be converted using a similar process. So that was with phase 1. Now with phase 2 reactions, we have what are known as conjugation reactions. And under conjugation reactions, we have got the role of glutathione, which will be attached to the pesticide molecules derived from phase 1 reactions. Now, the enzyme which facilitate conjugation reactions that involve glutathione, we have a glutathione S transferases, or GST. We have got the SH component, which is part of glutathione, and that is the reacting component. You could be interested to know that glutathione itself is made up of 3 amino acids, and we refer to it as a tripeptide molecule. So it's more or less like protein, which contain these 3 amino acids: glutamate, number 1, cysteine, number 2, and glycine, number 3. We can write it in shortcut like glue attached to cis attached to gly. Now we can have a simplified reaction which involves glutathione. And the glutathione, we can represent it by this structure, where the SH, the reacting component, is attached to cysteine, one of the amino acids. Where this is the herbicide carrying an OH, it will simply join at the sulfur position here and form this sort of structure. So we have a glutathione conjugate. And this reaction is catalyzed by glutathione S transferase. Now, glutathione conjugates could be transferred to the vacuole if it was in a plant. Now, let's suppose we have got this pesticide, which was an epoxide. That is, we have oxygen joined to it. Now, glutathione S transferase, but this one we can call it glutathione S epoxide transferase because it's a special transferase which is acting on this epoxide. So glutathione is going to get attached to this position here and detoxify this pesticide. We also have this 3,5-dinitrobenzene, which can conjugate with the glutathione. So we can have this structure being joined here to form this complex. Here we've got this pesticide diazinon. Now, diazinon can split here, and instead of having this structure, we add the glutathione there to detoxify that diazinon, which is an insecticide. Again, glutathione can combine with something like this methyl iodide, replace the iodine there with the glutathione via the S bond and liberate this iodine. Iodine is now hydrogen iodide. Probably we can use methyl parathione. Methyl parathione can split here. This bit can now combine with the glutathione. Once we have done that, this insecticide is no longer toxic. Glutathione can even combine with the atrazine via the chlorine atom and detoxify this atrazine. And of course, you have this complicated structure where you have glutathione attached to atrazine. And the glutathione S transferase can facilitate this reaction. Remember, this is part of detoxification processes taking place in a living system. Here we've got DDT, which is an insecticide. There's also a possibility of DDT combining with the glutathione at this position, also detoxifying it. We also have the propaclo. I think this is a herbicide which can also combine with the glutathione. Probably at this stage, I should emphasize that glutathione can combine with a lot of pesticides. Either it can combine with those pesticides carrying chlorine or iodine. Or it can combine with those insecticides which are in the organophosphate groups. So there are various pesticides which can combine with glutathione. I just want to move on to talk about other conjugating agents which are found in living systems. We have got glucuronic acid. It looks like glucose. Instead of having the alcohol, we have got the acid on the sugar molecule. Then we have got glucose, arginine, glutamic acid, glutamine, glycine, then sulfate, which can also play a role in various organisms in detoxifying pesticides. We'll look at some few examples. Here we want to look at the role of glucose and glucuronic acid. Glucose and glucuronic acid combine with some of the pesticide products formed in phase 1. The donor of glucuronic acid is uridine diphosphate glucuronic acid, UDPGA, but the donor of glucose is uridine diphosphate glucose, UDPG. This then is glucose. Focus on this structure where we have the OH. The only difference with the glucuronic acid is here, where we have an acid. So glucose, which plays a role in plants, glucuronic acid, which plays a role in animals. So let us talk about sugar conjugates. Here we have got herbicides, we can say pesticides. Pesticides, pesticide metabolites with the following functional groups are needed: the amine, the carboxylic acid, the phenols. So these will be able to form sugar conjugates. Uridine diphosphoglucose, UDPG, is the sugar donor. And UDP glucosyl transferase need UDP glucose as the sugar donor. Here is a simplified reaction which involves the herbicide metabolite and UDP glucose. So UDP glucose donates glucose to this herbicide product so that we form glucose or herbicide. And this is facilitated by an enzyme known as glucosyl transferase. Once glucose has been donated, UDP is liberated there. This can happen to, for example, Basagran. Basagran is carrying an OH here. We can combine this bit with the glucose coming from UDPG and form this complicated structure. And this is facilitated by UDP glycosyl transferase. Then we will have fixed the basagran. It's no longer active. We can also have the herbicide carrying an amine group. Then glucose, once again, is donated. Then we have a structure known as glucose N herbicide as a result of this reaction under the influence of glycosyl transferase. And the glucose is donated again by UDP glucose. Here we've got the insecticide known as the parathion. Now, UDP glucose can donate glucose to parathion because parathion has got this amino group. It is going to form an N bond to glucose after glycosylation. And this insecticide is no longer toxic. Now we can also have glucose ester formations, where we have probably the herbicide carrying the carboxyl group. It could be any other pesticide for that matter, which combines with the glucose, forms glucose CO herbicide or glucose CO pesticide. And we need glycosyl transferase to facilitate this reaction. Here we've got 2,4-D combining with sugar, forming glucose ester of 2,4-D. And then the herbicide is no longer toxic. We can also have, this is the Cabril. Now, Cabril, because we are having this process inside an animal, it can combine with the glucuronic acid to form this bit here. Or it can form the glucuronic acid via this bond here. If it was in plants, it was going to be attached to the glucose, but in animals or insects, carbaryl could be attached to the glucuronic acid. Here we've got the amino acid aspartate, which is combining with the herbicide carrying a COOH. Then we have a herbicide CO being attached to the aspartate. Again, here we've got 2,4-D being attached to aspartate. So 2,4-D can be destroyed by the amino acid. Here I've got sulfate conjugates, where we have this structure. So the sulfate, which can combine with the sulfate, which is known as PAPS in short, 3-phosphor adenosyl S-phosphate, or just call it PAPS in short. It can either combine with ROH, an amine, or it can combine with hydroxyl, sorry. It can combine with RNH2. It can combine with RNH, then OH, and form these structures. So this is another way of actually removing the pesticides using the sulfate conjugation. We can just simplify it like this. And the enzymes are known as sulfur transferases. I just want to move on to the phase 3 reactions. Now, plants do not excrete, let's say, pesticide. Let us generalize it. Pesticide metabolites. So further conjugation of phase 2 products is required. Then these conjugated products are now moved to the vacuoles for storage. Some of them can be used to be part of the cell wall. You can have pesticide glucosides. Let us call them pesticide glucosides to cover everything. Are further processed by the addition of malonic acid residue to the sugar conjugate. Of course, here we can use the glucose O herbicide formed in the phase 2. It can combine with malonate via malonyl coenzyme A and form malonyl glucose herbicide. And we release coenzyme A. I mean, this can be part of the cell wall, or it can be moved to the vacuoles. For example, we can have further conjugations. Let's say we had metribuzin glucose. We can also add glucose. Then we are having metribuzin glucose, which is a joint biocide. Or it could be just glucose added to pesticide glucose. Then we have pesticide glucose, which is also a joint biocide. So we have attempted to look at the breakdown of pesticides when they enter the living system. Remember, we have got so many of these pesticides, and we cannot follow each story. But at least where we have something which is common, We will try to get the generalized picture of what happens to pesticides when they enter the living system. So I will just give you a summary of pesticide metabolism. I know it has been a long journey from where we started and where we are now. Maybe we need to review what we have been talking about. I said if the pesticide enters a living system, it can be engaged or it can be confronted by various enzymes. Under phase one, we've got what we call hydrolysis, which can specialize on certain structures as follows. We've got carboxyl esterases, amidases, which specialize on certain structures. And these are some of the products which we can get. So we need to understand the role of hydrolysis. We also need to understand the role of Cytochrome P450, which is involved with oxidation, reduction, and hydrolysis. And what is happening to the pesticides is that they are becoming water-soluble and less toxic as they are being changed. And the cytochrome P450 is actually involved with some of these processes. And usually what we are doing is adding OH, NH2, SH, COOH. These are functional groups which are added to the pesticides. So this could be a summary of some of the things which are taking place, where if we have this benzene ring, we are adding an OH there. This is hydroxylation. We have O dealkylation, N dealkylation, oxidative deamination, sulfoxidation, nitrogen oxidation. This is reduction, where we are changing nitrates to amines. We're having hydrolysis of esters, hydrolysis of amides, hydrolysis of nitriles. Now, in phase 2, we were having the role of sugar, amino acid, glutathione. It could be also glucuronic acid. And you are having conjugation reactions with either glucose or glucuronic acid. And UDPG is actually donating glucose. In phase 3, we have a further alteration where we are adding more sugar to either malonate or malonyl pesticide glucose, where we'll end up with malonyl pesticide glucose. So basically, these are some of the reactions which we are making. Where we have glucose added to the herbicide product, or we have got maronite or maronate glucose added to this herbicide structure or pesticide structure. So, in a summary, this is how the pesticides can get detoxified. It's quite a lot of processes which are varied, which can take place in microorganisms, plants, and insects. So I will stop here. Hello?
Hello, Doc.
Yes, we have lost somebody, isn't it?
Yes.
I think it was too long, don't you think?
Yeah, sure, it was too long.
Yeah,
But I think she left just about 2 minutes ago.
okay. Anyway, I just wanted to cover everything under pesticide metabolism.
Okay.
Yeah, it looks like what we talked about when we were doing the herbicide selectivity. But during that time, we're just focusing on the herbicides. But the same principles can apply to all the pesticides.
Okay.
And using those principles, we can now understand why some pests are not killed by certain pesticides or why they are resistant to certain pesticides. It may depend on the metabolism which is occurring in those pests. Any questions?
Pertaining to the lecture, I don't have much of a question. I don't have any question. But
Anyway, what we will do, I think tomorrow we will talk about a bit on organophosphates and some fungicides. I mean, it may not be possible to cover all the pesticides, but we can just use some of the principles in order to understand how pesticides function, some of the problems which are caused by pesticides. Probably we will appreciate them if we understand some of these principles. So tomorrow I may focus on organophosphates and later on I will talk about the fungicides. In the future, we are aiming to talk about integrated pest management. Maybe I will have two more lectures under integrated pest management. And that should wind this course. And probably we may finish next week on Tuesday,
okay, Doc.
which means we will not meet on Saturday, is it?
If there's need for us to meet, you can meet on Saturday.
Eh?
If there's need for us to meet, we can meet on Saturday.
I thought probably during the weekend we may want to catch up on your assignments.
Oh, it's all right.
Yeah. During the weekend, try to catch up on your assignments so that I think next week we should be able to finish on time.
Okay, Doc.
Okay, thank you. We have lost one, isn't it? Okay, let me stop recording.